my screen is in full mode screen. Is it full screen?
Yeah. Right.
Perfect. Perfect. Thank you very much, Fawn. It's a great pleasure talking with you today. It's an honor to presenting my work here at Caltech. So today's talk is about network models for precision targeting of risk genes in brain diseases with a focus on schizophrenia and autism spectrum disorders, both at bulk tissue and also single cell resolution. Now, going over the outline of my talk today, first I'll be going over quickly on omnigenic hypothesis in studying complex disorders. And then I will briefly discuss actually the thread of research programs that I'm going to be talking about today. Then I will go over the first part of my talk, which is about network deconvolution and also how to drive master regulators from biological networks with a specific case of schizophrenia. Then I will bridge the first part of my talk by talking about cell specificity and protein dynamics to the second part of my talk, which is about integrative omics data modeling with the application in neuropsychiatric and neurodevelopmental disorders. And finally, I will talk about the concluding remarks and potential future avenues. Now, neuropsychiatric and neurodevelopmental disorders such as schizophrenia and autism spectrum disorders are known to be genetically heterogeneous and complex, and are usually defined as polygenic disorders. So with the advent of genome-wide association assays, as you mentioned, or GWAS assays, significant insights have been generated into the disease etiology, where in case of schizophrenia so far, over 250 common genetic variants have been found to be disease-associated. Now, in specific case of schizophrenia, 50% of the disease heritability has been defined, can be actually explained by common variants. Now, although remarkable, still critical questions remain unanswered. Now, GWAS hits are not necessarily disease causal and may act as proxies for pathogenic variants with which they are in linkage disequilibrium, or LD. For example, while most antipsychotic drugs exert their effects through blockade of type 2 dopaminergic receptors, or DRD2, which is a mechanism that was discovered over 50 years ago, no significant loci actually has been identified to directly associate with DRD2. While in polygenic approach, a handful of genetic factors give rise to a trait, recent hypotheses on omnigenic nature of some complex disorders basically have led to creation of a new research avenue to identify actionable core genes rather than focusing on a specific locus. Now, all these points basically prompted us to conduct a series of studies to leverage and develop computational models in order to address these challenges. Now, in this talk, first, I will go over parts of a cascade of multiple interconnected projects being conducted over the course of 3 years, starting with network deconvolution from post-mortem brain RNA-seq data to characterize transcriptional master regulators in brain disorders with applications in 4 diseases, including schizophrenia, post-traumatic stress disorder, or PTSD, autism spectrum disorders, and Alzheimer's disease. Next, I will briefly talk about our cell type deconvolution method called CellR, which is a novel method for characterizing cellular proportions in bulk libraries, as well as a meta-heuristic optimization model for inferring cell-specific expression profiles directly from bulk libraries, followed by creating a standardized pipeline for fine mapping and characterizing the functional impacts of genetic variants. And finally, I will focus on our recently developed model called MapSD, which stands for Markov affinity-based proteogenomic signal diffusion, which is a graph-based method leveraging graph signal processing fundamentals to identify unknown disease-associated risk genes in polygenic disorders. Now, I will be focusing mainly on the results of and findings of the first and the last project in this slide. Now, with this brief introduction, I would like to draw your attention to the first study on network deconvolution in order to identify transcriptional master regulators in complex brain diseases. In this study, given the fact that most of common variants associated with schizophrenia are of small effect sizes, we aim to potentially identify if a HOP gene within the regulatory network can act as the regulator of a large number of target genes, while contributing to basically to the disease pathogenesis. Now, starting with obtaining 2 sets of independent RNA-seq data from Common Mind Consortium, or CMC, and a second, a little bit smaller scale data from USC, which was basically generated from cultured neuronal cells derived from olfactory neuroepithelium, we created regulatory networks using mutual information as a measure of co-expression of genes, given that this type of actually measure is resistant against dimensionality reduction, and its power, it basically is powerful to preserve the geometric properties of the data points in lower dimensions. Now, this is a multi-step study, starting with numerical analysis and network modeling, up to the functional validations of the findings using IPS models. So if we start from panel A, clockwise, I will go over every single one of these steps. Now, upon creating the network in order to basically maximize the likelihood of keeping true positive edges, which is a well-known term, basically. Actually, we employed data processing inequality, or DPI, which is a well-known term in information theory, in order to trim the initially generated network. Next, we used the trained network and also the case control gene expression data using an improved version of a method called virtual inference of protein activity by enriched regular analysis, or VIPER, which used to be initially developed by our collaborators at Columbia Med School. Now, the ultimate goal for this process is to test for regulon enrichment. So here, by regulon, we mean the targets of a HOP gene within the regulatory network. So as I mentioned, the ultimate goal for this process is to test for regulon enrichment on gene expression signatures and to list transcriptional master regulators. Now, in this process, we first create the gene expression signatures for each highly connected gene, followed by computing its mode of regulation to specify what target genes are activated or repressed upon expression of the HOP gene within the network. Then we calculated the regulon enrichment for each HOP gene using a test called analytic rank-based enrichment analysis, which is also called AREA. Now, this will yield an activity score for each HOP gene, given its targets in the network. At the end, using a method developed by Lefort and colleagues, we corrected the computed activity scores for pleiotropy. So pleiotropic regulation of gene expression, which means basically genes being regulated by several different transcription factors, basically can lead to false positive results if a non-active regulator shares a significant proportion of its regulons with a bona fide activator, with a bona fide active regulator. Now, the outcome of this stage is a list of highly active master regulators for further downstream analysis. Now, conducting independent analysis both on the CMC consortium data, which contains, right now, I think it contains over 700 samples, post-mortem brain samples, and also the Xenon cohort, led us to identify 5 master regulators, which were basically among the top 10 hits in the both data sets. So this observation on independent data sets actually convinced us to take these genes for further analysis. Now, given that, so these 5 genes I'm showing you here at the top, including TCF4, NR1H2, and HDAC9. Now, given that TCF4 is known to be associated with Pitt-Hopkins syndrome, as well as intellectual disabilities, as well as that actually it harbors a GWAS loci in schizophrenia, we picked this gene for additional analysis. We checked whether the promoter region of the predicted TCF4 targets are enriched for TCF4 binding motif sequence. So for this, we conducted transcription factor binding enrichment analysis. As you can see on the left-hand side, we extracted the sequence of the target genes of TCF4, 2 kilobases upstream and 1 kilobase downstream of their transcription start site, and used a significance threshold of 80% to test the relative enrichment scores of its target genes. For this, we've picked an independent and non-relevant set of genes and ran JustPAR, which is a very well-known tool in the community. So we ran this on the targets of TCF4 in our network. And we found that basically the observed binding enrichment scores is significantly higher than expected by chance. So this indicates that numerically, TCF4 binding to its identified targets is at least in silico real. Now, to investigate the functional relevance of TCF4 dysregulation in schizophrenia, we examined how the predicted TCF4 transcription subnetwork changes with decreased TCF4 expression in iPS-derived neuronal cells. So we managed to differentiate neuronal progenitor cells to study the impact of TCF4 knockdown during neurodevelopment, as well as glutamatergic neurons. We ensured knockdown efficiency by real-time qPCR, as shown in the bottom. And then the generated samples were subject to bulk RNA-seq. Now, upon sequencing the generated samples, we obtained a list of course, differential expressed genes caused by TCF4 repression. We found that over 4,500 differential expressed genes are in NPCs or neuroprogenitor cells, and over 3,000 differential expressed genes in glutamatergic neurons, as shown on the top panel. Then we examined if the gene network perturbed by TCF4 knockdown in iPS-derived neurons is basically concordant with the predicted TCF4 regulons generated by the brain RNA-seq data with the targets that actually we generated from the CMC data. Now, our observed enrichment of transcription factor binding motif of TCF4 in its regulon were purely a DNA sequence-based prediction. So we next tested whether TCF4 can physically interact with its regulons. To do this, we first identified the target genes of TCF4 by examining the empirical transcription factor occupancy using ATAC-seq and ChIP-seq in neuronal and non-neuronal cells, as shown in the middle panel. While we observed significant enrichment, in fact, the enrichment level in neuronal cells was shown to be meaningfully higher than non-neuronal cells. Now, although for all these data sets we observed a significant overlap, but the enrichment score in glutamatergic neurons was meaningfully higher than the others, specifically neuroblastoma cell lines, which implicates importance of tissue-specific investigation of transcriptional regulation. Then we were interested to characterize what biological pathways might have been perturbed when TCF4 is repressed. In fact, we found that a large number of pathways relevant to neuronal activities are perturbed solely by TCF4. So this made us further conduct pathway enrichment analysis using Metacore. Some of these neuronal activity-related pathways, which were directly perturbed by TCF4, are long-term potentiation and also axon guidance. Now, the top gene expression changes in neuronal progenitor cells basically converged on early neurodevelopmental processes, such as axon guidance, neurogenesis, and attractive and repulsive receptors, which guide neuronal growth and axon targeting during neurodevelopment. In contrast, the most significant categories in glutamatergic neurons involved cell adhesion and cell matrix interactions, which suggests that TCF4 regulates different subsets of genes during neurodevelopment. In addition to these, we sought to investigate if TCF4-induced up or down-regulated genes cause distinct cellular functions. So we observed distinct functions in upregulated genes in neuronal progenitor cells versus glutamatergic neurons, while almost similar pathways were observed in downregulated genes. And finally, for both upregulated and downregulated genes by TCF4 knockdown, NPCs showed more enriched neuronal activity, actually gene pathways related to neural activity, than in glutamatergic neurons, with greater significance. So these results suggest that TCF4 gene targets act at the earliest stage of neurogenesis and may be more relevant to schizophrenia neurobiology. Now, although analysis of TCF4 knockdown in neuronal progenitor cells and glutamatergic neurons supported the role of TCF4 as a transcriptional master regulator, the TCF4 gene expression activity, which is the correlation between TCF4 expression and its regulons expression patterns, may be different in neuronal progenitor cells and glutamatergic neurons. So this cell type specific expression of the network activity may inform when the temporal expression of TCF4 is most relevant to the disease pathogenesis. Toward this end, we carried out simple gene set enrichment analysis, or GSEA, and the CMC data, as well as the TCF4 knockdown RNA-seq data in NPCs and glutamatergic neurons. Now, we found that the schizophrenia case control expression differences of TCF4 regulon in the CMC data, which was basically generated from dorsolateral prefrontal cortex in the brain, basically appeared to be more correlated with the TCF4 regulon expression changes upon TCF4 knockdown in NPCs than in glutamatergic neurons. Now, to demonstrate the closer similarity of the TCF4 interactome expression activity in the prefrontal cortex and in neuronal progenitor cells, but here we depicted actually this GSEA enrichment scores in 3 data sets. As shown, we have created this circular plot. In fact, we found that the pattern of the schizophrenia case control expression differences of TCF4 and its regulon in the prefrontal cortex of adult brains appear to be more similar to the patterns of higher TCF4 network expression activity in neuronal progenitor cells than in glutamatergic neurons. Now, one of the limitations of our observation is that both glutamatergic neurons and neuronal progenitor cells basically represent early neurodevelopmental stages, which basically would not recapitulate the actual gene network in prefrontal cortex in adult brains. Nevertheless, our observation is intriguing and may arguably imply that TCF4 may be a risk factor at the earliest stages of neurodevelopment and neurogenesis, which of course warrants further investigation. In addition to all these, we also intersected the GWAS hits from 2 credible resources, the PGC, or Psychiatric Genome Consortium, and also the CLOSA Consortium, and found that a large number of these genes are predominantly dysregulated in neuronal progenitor cells rather than glutamatergic neurons when we repress the expression of TCF4. Now, on this, we can conclude that TCF4 and its regulons in the earliest stage of neurodevelopment may be more relevant to schizophrenia pathogenesis. However, additional studies should be conducted to further validate this, of course, hypothesis. Now, with these findings and the effect of cell specificity on functions of transcriptional master regulators, I will go over the topic of cell specificity to connect the previous study with the next section. Now, in the previous study, while we characterized TCF4 as a major contributor to schizophrenia, specifically during neurodevelopment, we found that its mechanistic effects are remarkably different in neuronal progenitor cells, or NPCs, compared to glutamatergic neurons. On the other hand, computational methods designed for handling bulk RNA-seq data are not able to distinguish the source of signals extracted from the data since the libraries are a heterogeneous mixture of multiple cell types. Now, moreover, in a recent study, we also showed how differently the functional impacts of schizophrenia genetic variants are in different cell types. In addition to all these, a very valuable study by Skin and colleagues shows that in fact, schizophrenia common variants predominantly enrich for specific cells, including pyramidal and medium spiny neurons, while not being enriched for other neuronal and non-neuronal cells. Now, while we are well aware that how genetic risk factors can differently affect distinct tissues or cell types, still some critical questions remain unanswered. One of this is important point is proteomics and its dynamics. Now, in fact, amino acid research has been lagging behind nucleic acids, such as genome and transcriptome. This makes it even more critical when we know that protein abundances, as well as the type of proteins, whether they are secreted or membrane proteins, differ remarkably between tissues. Moreover, in addition to protein expression, the way the proteins localize in subcellular microdomains also poses novel challenges in integrated modeling of complex diseases such as schizophrenia. For instance, here on the right-hand side, which I've taken from the very important resource by Human Protein Atlas, which was published in late 2017, it shows basically how proteins, here marked in green, localize in various subcellular microdomains within each cell, and that how their trafficking patterns are different in distinct tissues, cell types. Now, to consider the points that I have mentioned so far, and to create a computational tool that can jointly model data from nucleic and amino acids, we need to address a number of immediate points to address, including cell specificity for disease of interest, cell and tissue-specific protein dynamics, and also the small effect size of common genetic variants, as well as polygenic nature of the diseases that where genetic risk factors may act as modules of a network rather than an individual locus. Therefore, in continuation of this thread of research, a vision is to create a computational tool for integrative modeling of interconnected data modalities to characterize unknown genetic drivers of a complex brain disorder. Now, with these short examples on cell specificity of genetic variants, I will go over the second half of this talk, which is basically about how we can integrate various types of data modalities at single cell resolution and model them using protein dynamics in order to characterize unannotated disease risk genes. Now, although proteins are treated as proxies of gene functions, multiple lines of evidence report a maximum of 60% correlation between the gene and protein expression levels only in certain organisms. Moreover, the functionality of proteins is not restricted to their abundances, where other determinants such as biochemical and physical properties, including subcellular localization, protein-protein interactions, and post-translational modifications also affect such functions. So this is the motivation of this research. Here we introduced Markov affinity-based proteogenomic signal diffusion, or MAPSD, which is a systems biology approach to identify novel risk genes, novel candidate risk genes in complex disorders. In fact, this method tries to model intracellular protein trafficking paradigms and also tissue-wide single-cell protein abundances at the same time. This method tries to integrate multiomics data aimed at amplifying the signals at risk loci with small effect sizes in order to reveal the convergent disease-associated genes modules in the brain. So without going into the mathematical details, just going over quickly on this, on the overall scheme of this method. Basically, initially, we have two main modules on the top, as you can see. The first module, we need to curate as much information, as much biological information as we can from the literature. And then using this information, we compact them and encapsulate them into a matrix that we call them signal matrix. In parallel with this, we have curated basically a large-scale protein-protein interaction network from multiple resources, where we had basically gone after highly accurate and highly reliable interactions, basically. Upon curating this protein-protein interaction network, we create the matrix affinity matrix, and then using the information from Human Protein Atlas, we use the subcellular localization from that atlas in order to adjust the network and its respective affinity matrix. So using this subcellular localization, we will be able to create tissue-specific or cell-specific protein-protein interaction networks. After creating this tissue-specific network, we will be able to, using the basically the Markovian properties of protein-protein interaction networks, we can basically propagate the initial signals that we have on known disease-free genes, as shown in the middle panel in red bars. We will be able to propagate this information throughout the entire network, and then calculate for each cell type or tissue of interest. We can calculate the amount of signals after diffusing the known signals onto the network, which are shown here in green bars. So each green bar, of course, graphically here, the longer each bar is, the more signal it's got, and the more susceptible we hypothesize it is to the disease. Now, in this study, in order to adjust the networks and also get more information actually about the protein dynamics, we use Human Protein Atlas. On the left panel, you can see that in this study, we had used 131 combinations of cell types and tissues. Here, each color basically represents a tissue, and the forks within each color represent their corresponding cell types. And in addition to different tissues and cell types, we had considered the number of proteins being expressed in over 30 subcellular microdomains within each cell in the human body, as you can see on the right-hand side. Now, a very important feature in any type of computational models is robustness of the model. So multiple types of robustness analysis actually was done in this study, but just as a simple example, I'm showing you how we were able actually to verify the robustness of the model. So we performed a series of tests to see if MAPSC is able to recover known disease risk genes in case they are not included in the training process of the model. Just as an example here, I have shown two schizophrenia genes. Here we removed these genes when training the model and then estimated their brain-specific signal intensities after running the MAPSC. So we expected the model to recover these known genes in the brain, since they are basically known with high signal intensities actually in the brain. For example, on the top, DGKZ is shown, which is a well-studied schizophrenia risk genes, demonstrated to be differentially expressed, differentially methylated, as well as harboring GWAS hits and de novo mutation schizophrenia. In fact, MAPSC signal intensities for this gene are the highest in 3 regions, including neuronal cells in cerebral cortex, Purkinje cells in cerebellum, and neuronal cells in caudate. Another example is ST8, SI82, and also there are multiple other examples that, if you're interested to know more, please feel free to check the paper and discover more about them. Now, in addition to schizophrenia, we were also interested to apply MAPC to autism spectrum disorders to potentially identify novel risk genes. Just to give you a quick description of autism spectrum disorders are a group of neurodevelopmental diseases predominantly characterized with behavioral and communication impairments. In the US, according to the latest reports that I was reading, almost 1 out of 59 children are diagnosed with autism across the US. So that was a good motivation to focus on this disease. A very important point about autism, if we want to use MAPC, is that we need to work on those type of complex diseases that we have enough information to train the model. So this model requires a lot of information, and that's why autism is a very good candidate to study, because there are quite a lot of genetic information and a lot of GWAS studies and exome and whole genome sequencing studies on autism spectrum disorders. So in this study, we had collected 3 layers of omics data, including genome, transcriptome, and epigenome. Upon running the model, we came up with a short list of candidate risk genes. We were interested basically to analyze the spatial enrichment of our finding within a cell at the protein level and then compare them with the known autism genes. Among known autism risk genes, we observed that 62% were highly expressed in neuronal cells within cerebral cortex, as shown here on the left panel, while in total 80% of the entire gene sets were highly expressed in various cell types in cerebral cortex. Now, among candidate genes generated by MAPC, 75% also were highly expressed in neuronal cells within cerebral cortex. And we observed that actually also 63% of the total number of newly identified gene sets were highly expressed only in cerebral cortex. So this is strong similarity on a spatial distribution of the expression of the known and also the identified candidate risk genes actually prompted us to do additional analysis in the next slides. I should mention that actually this figure is generated at protein level, not at gene expression level. In addition to this, we're interested in finding where protein products encoded by autism risk genes localize in subcellular microdomains. First, we checked the autism genes with the highest MAPC signals in the brain, and we observed significant enrichment of the disease susceptibility risk genes in neuronal cells within cerebral cortex, as shown on the left-hand side. In fact, 82% of these loci were enriched in different cell types across cerebral cortex. And among them, 5 subcellular microdomains harbored over 55% of the entire loci, essentially enriched in neuronal cells, including cytosol and nucleoplasm. So this is an indication that not all of the domains within a cell are disrupted by the disease equally. Similarly, in the newly identified gene set, we observed that almost 80% of the identified genes were enriched across various cell types within cerebral cortex. And we did not observe any significant difference between the proportions of enrichment percentage in cytosol, nucleoplasm, nucleus, plasma membrane, and vesicles. So there's a good concordance actually between the spatial enrichment of our findings and known autism genes. Now, because autism, as I mentioned, is a neurodevelopmental disorder, we next investigated if the brain-specific MAPC genes are dysregulated during various developmental stages in human brain. So using Atlas of Developing Human Brain, or BrainSpan, on 16 brain regions, we divided the data into 2 large categories of prenatal and postnatal stages, where each with various time points. Where, for example, the prenatal stage includes 0 to 12 post-conception weeks, or PCW, 13 to 24 PCW, and 25 to 36 PCW. Then we averaged the expression levels of each MAPC genes across different stages of prenatal and postnatal stages and looked for differential expression. In fact, we found that almost 94% of the known autism genes, as well as 86% of the identified candidate genes, are differentially expressed in postnatal stages versus prenatal stage. Now, the overall pattern of the number of DE genes or differentially expressed genes in autism and MAPS genes were also similar. Now, to gain further evidence supporting their disease relevance of our findings, we analyzed the tissue-specific expression levels of the identified autism genes at mRNA level. For this analysis, we used the gene expression levels on 53 different tissues from the GTEx consortium. Of course, this is a version 7 in that time, but I think version 8 has already been published. Now, just to tell you what GTEx is probably, is that GTEx data basically contains mRNA levels across the entire transcriptome, which enables us to specify to what extent a gene is expressed in distinct tissues. Now, we observed a strong similarity in the expression profiles of the identified risk genes with known autism genes in the GTEx data, as you can see on the right panel. Then we looked for demonstrating if any of the identified genes are specific markers of brain cells in post-mortem adult brains. For this, we used a very good resource of single-cell data generated in the brain by Lake and colleagues, which covers over 60,000 single cells from frontal cortex, visual cortex, and cerebellum. This data includes 7 cell types, such as excitatory and inhibitory neurons, microglia, and endothelial cells. In fact, we found that all of the MAPS genes with the highest signal in the brain are highly enriched in all of these sets. Yet the enrichment degrees in neuronal cells, such as inhibitory neurons, was apparently higher than non-neuronal cells, such as OPCs, or oligodendrocyte precursor cells. So this observation provides a strong evidence on how certain cell types are involved in the disease and what gene markers basically are perturbed during neurodevelopment. In addition to work on autism, we also conducted a similar study on schizophrenia and found strong similarities basically between the known disease genes and the novel candidate genes in terms of expression patterns at distinct tissues, as well as their expression profiles during neurodevelopment. Now, one of the major challenges we were facing in this study was basically to demonstrate how our findings actually make sense in real world, because it was purely numerical. So we were basically lucky to see this paper published at the same time by Velmeshev and colleagues, which is currently the largest single cell data on post-mortem autism brains. So we intersected our top candidate risk genes with a list of cell-specific dysregulated genes in their paper. By this, we actually we made interesting findings where we found that 21 candidate genes, which was basically among the top 30, I think, MAPS hits, are in fact dysregulated in specific cell types. And these genes have not been identified before to be dysregulated in autism. And no bulk resolution RNA-seq or any type of similar assays had not actually revealed these genes to be dysregulated in autism. Here as an example, I'm showing 4 of these genes, including EMX2 and KCNJ3, that we had found to be disease-associated in subgroups of neuronal cells, as well as astrocytes. Now, with this single-cell project, we found that, in fact, our top identified hits are really dysregulated by disease, and the model and the major hits, the major outcomes of the model are basically accurate and reliable. Later, we focused on these candidate risk genes and mentioned, as I mentioned earlier, so we checked if there are any loss of function mutations in any of these genes, whether they can lead to any autistic phenotypes in mice. So For doing this, we leveraged mouse genome informatics database, and we observed hyperactivity and hypoactivity, as well as impaired motor capabilities as a relatively consistent behavioral pattern among these knockout mice, while abnormal neuron physiology was shared predominantly as a common phenotype in the nervous system. In addition to that, we sought to investigate if any of these genes are associated with any Mendelian diseases. Of course, we use OMIM, or Online Mendelian Inheritance in Man, database, and found that 5 of these genes are actually associated with some known Mendelian disorders, including seizures or certain types of epilepsy. Now with this, and for the interest of time, I would like to move to the conclusion part. So in this presentation, basically, I talked about our research on how we discover TCF4 as a transcriptional master regulator, and that how its function is basically cell-specific, and basically how it is related to schizophrenia, mainly during neurodevelopment. Then I reviewed our work on cell-to deconvolution, followed by a new computational method that enables us for jointly modeling proteome dynamics and the expression along with the other types of molecular data aimed at annotating unknown disease risk genes. Now, based on these results, there are a number of topics which are currently under investigation, including how we can develop tools for jointly modeling single-cell RNA-seq data with single-cell binding patterns of transcriptional factors to their targets upon projecting onto the regulatory networks to find cell-specific disease master regulators. The other question is that how we can use single-cell RNA-seq data as a proxy for proteome expression to quantify cell-cell interactions. And finally, how we can leverage the hidden knowledge in regulatory networks as well as genome-wide genetic aberrations, basically to create some sort of a risk score, as we call it, polygenic expression risk score, which basically has a potential to be of great clinical utility. The reason for this actually point that I'm talking is that basically most of the large-scale genome-wide studies are generated from European ancestries. However, there are quite a lot of other gene expression data which are generated from non-European ancestries, such as African ancestries. And this makes it actually easier for us to also apply our methods on some sort of populations beside European ancestry. And on the other side, in currently available polygenic risk scores, we are dealing with hundreds of thousands or sometimes millions of SNPs, which makes the models quite complicated. But in this case, if we can actually develop something on networks and at the same time focus at a single cell resolution, we should be able to create some sort of an expression risk score that can actually replace the current polygenic risk scores. However, this specific model is currently in progress. Now, with that, I would like to thank Dr. Fan for inviting me. It's a great honor. I would like to also thank all the people that have been involved in these studies from across the country, from Penn, from UC Santa Barbara, from Harvard, from Jefferson, from University of Chicago, Columbia, Northwestern, and also University of Southern California. With this, I would like to thank you very much for your